Hi, and welcome back to the Sound of Science. Today we're going to be talking about genetics, how we learned about genetics. We're going to talk about this guy, Gregor Mendel, and we're going to talk about how we can predict the offspring that are possible. So, the first thing, of course, that we're going to talk about is Gregor Mendel. And so, the question I have here, sort of jokingly, is he a brilliant scientist or a man obsessed with peas? And really, the reality is he's not either one. Gregor Mendel was a priest and he worked as a teacher out of his monastery, and he was also the head gardener. So he's in charge of growing all of the food for the monastery. And Mendel really was good at picking up patterns. And so when he is growing peas in specific, he starts to notice some things. First of all, peas have lots of traits that are only in two forms. For instance, humans come in a whole range of heights, right? We have some that are tall, some that are like a little tall, some that are super tall, some that are medium tall, some that are not quite so tall, some that are short, some that are super short, and so on and so forth. Mendel noticed that peas were either super tall or super short. There was no in between. Um, and there were like 8 different traits that met with that, including things like flower color and where the pods for the peas were placed, and whether those peas were yellow or green, or whether the pods were constricted around the peas or sort of puffed out. So, all of these different traits, Mendel started to notice that either it was one way or the other. In addition, peas were easy to work with because it was easy to grow and they were easy to control their reproduction. Peas don't care who they breed with. In addition, peas also produce hundreds of offspring, which made it a lot easier to identify these patterns when you're working with a number of offspring that are statistically significant. And of course, by offspring, we just mean kids. So let's talk about Mendel's experiment. Let's talk about Mendel's experiment. Essentially, Mendel took tall pea plants that had only ever produced tall offspring and who had come from a long line of tall pea plants, and he bred them with short pea plants that had come from a long line of short pea plants. He called these purebreds because they were always tall or always short. And this first generation of his, we're going to call the pea generation or the parent generation. So he bred this tall pea plant to this short pea plant. And I think Mendel kind of half expected to get medium pea plants, even though he'd never seen one before. But he'd also never bred a tall and a short together. So anything was possible. In that second generation, which we actually call the F1 generation or first filial generation, Mendel had all tall plants, which seemed a little bizarre to him. So he allowed those tall plants to fertilize themselves and fertilize each other. So all these tall plants from the F1 generation bred together. And in the F2 generation, their children, he had 75% tall and 25% short. So not only did he not get what he expected in the F1 generation, but one of the traits that had completely disappeared returned in the F2 generation. So Mendel continued to run this experiment over and over again, and not just looking at the height of the pea plant. He looked at those 7 other traits that appeared in these two forms. And from this, Mendel's able to get a pretty good idea of what's going on. Okay, so Mendel figures out that there has to be some factor that determines your characteristics. And he figures out that those factors get passed from parent to offspring, from parents to kids. He also figures out that whatever that factor is, it comes in different forms. So he names the factors the genes, and he calls the forms or the type, the variety, whether it's tall or short, the allele. So alleles are basically just saying this is the variety of gene that we have. The third thing he figured out was that somehow, some way, some of these alleles had to be dominant over the others, because sometimes alleles were able to show up, and other times that type of gene was hidden by another type. And so Mendel's starting to piece all of this together. He also realized that when gametes are forming, that there has to be some way for the alleles to separate. And therefore, only half of the gametes from one parent get passed on, so that when both parents, when their gametes join together, you get a full cell. So here comes the sciencey stuff. Mendel actually creates 4 postulates, and out of those we get 2 laws of inheritance. So postulate 1 is the principle of paired factors. Just says what he already learned. You have 2 factors that determine your genetic makeup. One comes from mom, one comes from dad. They come together, that gives you your factors. Postulate 2 is the principle of dominance. Sometimes one of these factors is dominant over the other, and so it may hide the other factor, so it appears that there's only that one. Postulate 3, the law of segregation or law of purity of gametes. This is Mendel's first law of inheritance. All this is saying is that the alleles have to be randomly separated into their gametes during gamete formation. This is essentially meiosis. This is what we see with meiosis, how the factors can separate into each of the 4 gametes. And postulate 4, or the law of independent assortment, this is Mendel's second law of inheritance. The factors for different traits are going to be sorted independently of each other. And I'm going to explain this one because this is the one that seems a little bit more confusing. So essentially what we are saying here is that if you have a gene for your hair type and a gene for eye color, they're not dependent on each other. You don't get brown hair just because you have brown eyes. They aren't related to each other, so that you could end up with different eye color or different hair color, and it wouldn't, those factors aren't connected to each other. Okay, my friends, we are going to move into talking about Punnett squares. Probability, the likelihood that an event will occur. This is not going to be a guarantee. It's just, okay, it's 25% likely that this will happen, or 50% likely that this will happen. There's still no guarantee. The phenotype is the physical appearance that you happen to see, the trait, what it looks like on the outside, or what characteristic it might have. The genotype are going to be the genes that are there that cause that physical trait. Homozygous, homo means same. It means the two alleles that have come together, those 2 factors, are the same factor. So you got the same allele from mom and the same allele from dad, okay? Heterozygous means the 2 alleles are different. Hetero means different, right? So in this case, you're going to get one allele from mom, but a different allele from dad, a different variety of gene from dad. And again, Punnett square is just a way for us to see the probability of certain offspring. Okay, a little bit extra notes. Homozygous is also the same thing as saying purebred. And the letters that come after that, that's where we're talking about genotypes. So you guys know that you have 2 factors, right? One factor from mom, one factor from dad. So if it's written as a capital, that means it's dominant. If it's written as a lowercase, that means it's recessive. So the dominant alleles are always going to show up if they're present. The only time we're going to see a recessive allele is if we have 2 recessive alleles. So what we're seeing right here is the genotype. So this is the genotype. It has 2 dominant alleles. That means this is homozygous, and it's also going to have whatever the dominant trait is. This right here is recessive. These 2 alleles are both recessive, and so it has the recessive trait. So this is how I like to explain it. These 2 big alleles, and we'll say this is for height of the pea plant. That's what these genes are for. The dominant one is like, be tall. So when these 2 alleles are talking to each other, this first one is going to say, I want the plant tall. And the second one is going to be like, yeah, tall's good. And so with that, you're going to end up with a tall plant, right? Makes sense. These guys, on the other hand, are a little bit weaker. And so this little one is going to be like, I like a short plant. And its friend over here, its paired factor, is also a recessive allele and also wants the plant to be short. So it's going to be like, yeah, I agree, short's good. And so because they're in agreement, we're going to end up with a short pea plant. Remember, dominant is an uppercase, lowercase stands for recessive. So then here is the big question. What happens when they're not in agreement? Well, when it's a hybrid or a heterozygous, it's actually going to go up something like this. Your dominant allele is going to come in and is going to be like, yeah, tall pea plants. And your recessive allele, this little lowercase, is going to come in and just kind of go, we could be short. And when you compare the two, which one are you likely to hear? Well, the dominant allele. That's the one that's going to make the most noise. So the end result is a tall pea plant. Even though this little short allele is here, it's not going to show up because it's being hidden by your dominant allele. So what we just talked about, if you have the recessive trait, you can only have the two recessive alleles. You can't have a dominant allele and have a recessive trait. If you have a dominant trait, you are either homozygous dominant or heterozygous. And if you are heterozygous, you have to be dominant. You don't have a choice. By definition, you have one of each. So you have to be dominant. So let's give this a little bit of a practice. What we're seeing up here is that the agouti, which is the same thing as a tabby stripe, is our dominant trait. And short hair is going to be dominant over long hair. So if we take a look at it this way, and we look at this cat right here, this cat is an agouti. It's got the stripes. You can see the stripes right there, right? And it happens to have fairly short hair. We're going to go with short hair on this one. I know it's a little bit longer than some cats, but we're going to go with short hair on this cat. So what we're saying is that this cat, its phenotype is short-haired agouti. Both of those are dominant. So that means that its genotype has to be dominant as well. So the options are for the agouti, for the tabby stripes, big A, or it could be big A, little a. Either one of them is going to give us the dominant alleles that we see in this cat. Same thing down here. Big S, little s, or big S is going to give us short hair because that's the dominant trait. So when we look at this cat over here, this cat very clearly has long fur and it has no stripes. So it's actually going to be recessive for both traits. So because this cat is recessive, we're going to see the recessive genotype. Remember, the recessive phenotype means a recessive genotype. Little a means non-tabby. Little s means long hair. So let's take a look at this cat up here. This cat is indicating no tabby stripes, right? And it seems to have short hair because remember, those two alleles are not connected to each other. They sort independently. So what we're seeing with this is a recessive phenotype for agouti and a dominant phenotype for hair length. So to make recessive, you have to use two recessive alleles. So little a, little a. To make a dominant, you have the option of either big S, or big S, little s. And we're going to see that that is what the genotype is for this cat. Genotype is going to be the genes that are present. Phenotype is going to be the physical appearance. Now it's time for us to look at these Punnett squares. And specifically, this Punnett square we're going to call a monohybrid cross, which is what you're seeing right here. So a monohybrid, mono means one, hybrid is just cross. So a monohybrid cross, like we see here, this is all about one allele at a time, or looking at one gene at a time, the two different alleles. And essentially, when you set this up, you're going to set it up by putting your parents at the top and on the side. And we're going to use those to determine the offspring that are possible in each scenario. So let me just clear this out and let's take a look. When we do Punnett squares, like I said, we're going to take our parents. So in this case, our parents are big T, and it's going to cross with a genotype that's little t, little t. So a homozygous dominant is crossing with a homozygous recessive. So, and if you take a look, this parent actually goes here. This parent actually goes here. So you can reverse these. You can put either parent on either side, but this is kind of how it works. So once you get your parents in place, you're literally going to just drop letters down and slide letters over. So if we drop down the big T, we're going to have big T and slide over the little t. Again, we would do the same thing here. Drop down the big T, and we're going to slide the little t all the way over to this box. So again, big T, little t. Again, drop down a big T, slide over that little t. Final box, drop down the big T, and slide over that little t. Okay, now that I've cleared it. So you can see in this Punnett square over here, what we had just seen over here with our big T's dropping down, our little t's sliding over. So the other thing to understand about this is that we are looking at percentages. Each one of these squares is worth 25%. Okay? So each square is worth 25%. Think of quarters. So 4 quarters makes 100, right? 100 pennies or $1. So when we are looking at this, every one of these that we see happens to be dominant. So that's 25, 50, 75, 100. So in this case, we've got 100% dominant phenotypes. Now let's practice. So we're looking at sneakiness in snakes. So sneakiness, we said, in snakes is going to be dominant. So sneakiness in snakes is dominant. Our first example is a heterozygous. So we're going to underline that because that's important. Heterozygous sneaky snake. And it's going to cross with a homozygous sneaky snake. So heterozygous means two different. So to make a heterozygous snake, and I'm just going to write it down here, it's going to have to be a big and a little. That's hetero. Homozygous means two of the same. And in this case, we've said that it's a sneaky snake. So if it's a sneaky snake, we know that it has to be dominant because sneakiness is dominant in snakes. So that means that our second parent has to be big, big. So we're going to place these parents, well, really where they go on the Punnett square. So we're going to put one on top, and we're going to take this other one, we're going to put it on the side, and we're going to do our drop and slide, which is what you see here. So big S, little s on the top, big, big on the side, and now we're going to drop and slide into the Punnett square. Big drops down, big slides over. A little drops down, a big slides over. And you might notice that even though I always say drop and slide, we're going to put the capital letter first, if there is one, because that's just the way that we do it. It's not wrong the other way, but the other scientists might laugh at you, so definitely don't do it that way. Okay? And then a big S, and then a big S, little s. So what you're seeing here is an example of, let's see here if I can get my pen in the right spot, 50% homozygous dominant and 50% heterozygous. So this is going to be 100% sneaky snakes. So let's try the next one. So in our next one, homozygous sneaky snake is going to cross with a homozygous non-sneaky snake. So again, we have some important words here. Homozygous. Homo means same. So a homozygous sneaky snake, if sneakiness is dominant, has to be big, big. If it's crossing with a homozygous non-sneaky snake, then we know this is recessive. So because this is recessive, that means it has to be crossing with too little. So too big crossing with too little. So let's take a look. There's our homozygous sneaky parent, our homozygous non-sneaky parent, and as we drop and slide and we fill in the offspring, again, we see 100% dominant. Only in this case, they're all heterozygous. So we have one more example. And this time I'm not going to underline things, but it's 2 heterozygous sneaky snakes. Remember, heterozygous means one big, one little, 2 different. So we're going to put the parents where they belong on the top and the side. And now we're going to drop and slide. And into our first box, we're going to drop down a big S and slide over a big S. So we have a homozygous dominant offspring. Drop a little, slide over a big, drop a big and slide over a little, and then finally 2 little. Over here, what we have is 1 dominant. So I'm just going to write a D here for dominant. Another dominant. This one, again, it has a dominant allele. It has to be dominant. And this one finally is going to be recessive. So we have 1, 2, 3, or 25, 50, 75% dominant. And here we've got our last one, 25% recessive. So you see how in each one of these we have different percentages. And so that's why it's important to always do your Punnett square. We are going to stop here. The dihybrid cross is a little bit more complicated, and I'd like to tackle that in the next video. So for right now, this is all I have. As always, thanks for watching.